they don't come apart in the right way, what happens? What will happen to those gametes?
You won't get necessary stuff that they need.
The necessary stuff what they need being what?
Being 23.
Right, exactly, Christina, the right chromosome number. So if they don't disjoin the right way, you're going to have the wrong chromosome number. You're going to have aneuploidy, which can likely lead to miscarriage. And miscarriage in humans is very, very common, actually. 1 out of every 4 pregnancies results in miscarriage. 1 out of 4. A lot of women don't know because it happens very early and they think it's a heavy menstrual flow. Yes, we'll talk more about that. So this a lot of times is linked specifically to these features that happen during meiosis, how the chromosomes split apart, whether they exchanged, recombined, did they disjoin the right way, and if they didn't, they have aneuploidy. That a lot of times triggers the embryo and the woman's body to get rid of the developing embryo because it has the wrong ploidy, okay? I don't know if this is, I'll skip this, it's just a little short, it's a little meiosis dividing the cells, chromosomes going, I don't know if it's gonna play. Sometimes I have problems with this movie. Is it playing? It acts like it's playing, but it doesn't do anything. All right, I'll show you later. I know, you guys are, wait till we get to the pregnancy and it's puberty, you know, so it's, a lot of those things are gonna blow your mind. So, can a differentiated cell be reprogrammed or differentiated? So that was meiosis. You have a germ cell and it goes down meiosis and it becomes sperm, core sperm. Or you have a germ cell in the early ovary in the fetus and it goes down and becomes one egg with a few polar bodies, okay? Those are differentiating, those are turning into very unique specific cells. Egg cells are not like any other cells in the body. Sperm cells are not like any other cells in the body. Just like skin cells or liver or intestine or hair or all these other different types of cells that you have in your body. Those are totally differentiated. So can a differentiated cell in its adult form, like an egg or a sperm, can it be reprogrammed or undifferentiated? Can we reverse the clock, the developmental clock? Can I take a skin cell and turn back time and get it to not be a skin cell, get it to be like an embryo-like cell or like an egg-like cell? Simone's saying yes. No. Yes? No? Split all over the place.
I would say probably.
Probably. Okay, well, internally you probably think, no, that's crazy. Well, that's what I used to think. Okay, that's what I thought, no, that's impossible, you can't do that. Like, when something differentiates, it's months to years of going down a path and becoming something that it now has a unique anatomy and physiology for. So if you told me, oh yeah, you can reverse and turn a skin cell into an embryo cell, I'd say, no, you can't. Well, okay, science, this is where science is very cool. It unveils these really amazing findings where we realize that what once we always thought was one way, we can actually reverse it. So I don't know if you guys remember, we talked about stem cells last week, right? And stem cells now, there's a new thing called iPS cells, or induced pluripotent stem cells. You can take a skin cell biopsy, a little core punch of skin cells, in the dish with a few different factors, we can turn those into embryo-like stem cells, ES cells or iPS cells, okay? That is basically reversing those and having them go back in their developmental journey. So basically, you have to switch gene expression from one cell type to another. You have to cause that cell to stop expressing the DNA of the genes it was expressing and start expressing new ones to give it a new identity. Or you have to switch from a differentiated specialized cell type to a developmentally more primitive or pluripotent state, back to an earlier type cell, which we would call pluripotent. Pluripotent means it can give rise to lots of different cells. So it's got, I will say, this is a weird way to say it, but it's got plural, like multiple, potential. And an embryo has plural potential or pluripotency or multipotency, and that can give rise to a lot of different cells, okay? So we would think, going through this, we've got the gametes that get made by meiosis, they give rise to the zygote, and then it gets a 2-cell, 4-cell embryo, blastocyst, embryosis about day 4, day 5, and then the later embryo, and then the fetus gets more of the proportions of the human, infant, baby, adult, elderly. So at this point, we say that the zygote and the 2 and 4 cell members, parts of the embryo, are totipotent. That means they have the total potential to give rise to every single cell in your body. And I know I teach this, and a lot of times people are like, oh yeah, totipotent, okay, embryo, 2-cell, 4-cell, they're totipotent, they give rise to everything. That is absolutely amazing if you think about it. You can start from 1 cell or 2 cells, and I can get every single other tissue, organ system, structure to make up a brand new individual. But they're only totipotent here. We start getting further away and we get to the later stage embryo. This is the one typically that will implant into the uterus. This is pluripotent. That means it's got the pleural potential and give rise to a lot of different things, but not everything. Not the placenta, not all the other structures. This can give rise to everything, placenta and the human and the person. So, can you reverse a cell? Well, we've already talked about iPS cells, or induced pluripotent stem cells, and we now know that development going down a certain developmental trajectory and differentiating, it can be reversed. That blew people's minds away. Starting in the 50s, they were cloning frogs. And then later in the 80s and the 90s, they started trying to clone mammals. And I'll show you exactly how that's done. Cloning basically turns one cell type into another cell type and sends it down a totally different trajectory. So we call this reproductive cloning or somatic cell nuclear transfer. Somatic cell means a body cell, like a skin cell. Like in this case, it's a somatic, like a body, a skin cell. Somatic cell nuclear transfer, or SCNT. And this is done, you guys are probably well aware of this situation with cloning mammals even now. When you were all born, probably about 1997, most of you were born somewhere around there?
99.
99, sorry, 99. Yeah, that's true, 97 is a little bit before your time, around the same generation. Dolly the sheep was cloned, remember? So you heard about it later. Well, I was in college then. It was like, oh, they cloned a mammal, they cloned a sheep, oh my God, a baby sheep. What they did is they took a skin cell from a sheep, just a regular sheep grazing, doing her thing. They took a skin cell. They took the nucleus, the 2N nucleus that has the full chromosome complement. It's not a gamete, it's not a haploid, it's 2N, okay? And they took an egg, oocyte, and they enucleated it. They sucked out the nucleus. So now you have the egg that underwent meiosis and did all that, but they took out the nucleus. They put in the adult skin cell from the adult sheep. They put that nucleus in the enucleated egg, or the egg that had its nucleus removed. This was triggered, usually you can give a little bit of electrical poration or a little bit of electrical current, and you can sort of trigger this. This will start dividing. Because it's 2N, it has the full chromosome complement. It's now in the egg, and the egg is ready to reprogram and allow the developmental embryo program. Cell divides, you get a blastocyst, you get an early embryo. You can implant that early embryo into a surrogate mom. You give them hormones, like a surrogate human mom. You just implant right into the uterus. This will implant, make a placenta grow, and then what derives from that is a little baby that was born. In this case, it was Dolly the sheep, cute little baby Dolly. That was her surrogate mommy. Cloning of the first mammal. We had already cloned frogs and chicks and all kinds of other things, but this is the first mammal. This was in 1997. This is showing that you can reprogram an adult cell and have it go backwards developmentally in time. And you can technically clone an individual even if you have a skin cell. Okay, but I will say, people, this is where we get, is this gonna go to humans? Are we gonna clone humans? Is this the next? Because we can clone mammals. I will tell you, even in primates, non-human primates, like macaque monkeys, the cute little macaque monkeys, they are cloned. Now we've been able to clone them, but it took a long time, thousands, tens of thousands of eggs and attempts to do this. Humans and non-human primates have things about their eggs in this cloning process that is very difficult. They're not easy, they're not simple, it's very complex. And so it's wasteful, there's bioethical concerns, and now even in the US and around the country, in different countries around the world, you can't even culture like an embryo in the dish for longer than 10 days. You can't, there's a 10 or 11 days. You can't even culture an embryo. So you can only study and do things in the very early stages. And I will say, even cloning in mammals, everybody's really excited about this. The genes are one thing. So the actual genes that you pass on are the chromosomes, right? But on top of the genes, on the chromosomes, there are different chemical marks. Enzymes go along and they mark different genes in different ways. We call it epigenetics. We'll talk about this a little more later. But regular genetics is changes to the genome, the DNA. Epigenetics is just little marks that are on those. Well, when you do cloning, a lot of those marks are erased and they're not there. So Dolly actually died very, very young. She had all kinds of issues. She had pulmonary, I think cystic fibrosis, respiratory problems, some liver problems, arthritis. Longevity was greatly decreased, so she had a higher mortality rate, didn't live as long, had all kinds of problems. Even though she was a clone, she came from another sheep. Epigenetic-wise, and this process of cloning leads to problems. So for me, human beings are never going to, I don't want to say never, but there's a lot of problems and there's so many little marks that we don't have worked out. And even to clone a mammal like this, I think she lived like 3 or 4 years. The average sheep lives 2 or 3 times that long. So there are ailments and diseases and stuff that come along with this process. And primates and humans are harder than that. They're even harder because there's more complexities involved. So this is just sort of going into problems that can happen in chromosome segregation. So that was a little segue into cloning, reproductive cloning, using an egg, taking out the nucleus, and then using just an adult cell. Some people will say, oh yeah, we're getting close, we're going to do human cloning, we're going to use CRISPR, we're going to do DNA editing, we're going to edit out diseases and different things. There is going to be a huge lag and oftentimes many ethical and biological obstacles that are going to make that very hard. And that's why I usually tell, we're not right there yet. There's, even in mammals and primates, there's still too many problems. Human beings not going to be willing to do that with all those kinds of complications and problems. Yeah, Christina.
So if you were to clone like something, are they gonna have the same DNA as the person that you're
Yep,
cloning?
exactly the same.
Like
That is the definition of clone. It's almost like mitosis. Just
so
my
like that blueprint, like that's
exactly the same. It is like an identical twin, but now in a different phase of time. So if I were to get a clone of me, have the baby, it would be me. It would be me.
you would get the baby.
I could carry it even. Weird. But I would not, scientifically speaking, there's too far to go. There's too many complications. There's too many areas that we don't have worked out. So good question, because the DNA is the same, but the epigenetics are not going to likely be the same. So the marks, the little caps that certain enzymes put on those genes to allow those genes to be expressed or turned off, those are not the same. They've already done this. We've done this in mice. We've done it in sheep. We've done it in cow. We've done it in all kinds of animals. And they always have some sort of diseases or ailments and aging problems. They age faster, ultimately, and they die earlier, right? Interesting, though, if you think about that little experiment. What about, like, raising yourself? It could answer all.
So
It's crazy, right? But it would answer things about, like, what's genetics versus nurture? What's the social? What's the genetic? Like, is it going to be just like me or raised different? It's going to be totally different.
Oh my
Who knows? Who knows? This
But
gets
then you
off
would, like,
into
see
another.
your younger self when you grow.
Weird, right? Yeah,
Like-
weird. But there's lots of issues.
And then you're like,
All right,
just
I gotta
look
get back
at
because
yourself.
I gotta finish
Look
this
at
stuff.
yourself
But
in the mirror. There
yes,
you
it's
go.
strange. Non-disjunction, if this happens, it can result in syndromes where the individual is aneuploidy. It has an incorrect number of chromosomes. So this is now talking about reproductive cloning. This is when chromosomes non-disjoint. They don't undergo disjunction. So normal meiosis is here, right? Now, when something happens where in meiosis 1, the chromosome didn't get pulled apart. So you've got 2 in one and 0 in the other, okay? And this is coming time to get fertilized. If it gets fertilized by a normal sperm carrying an X or a Y, it's actually going to be 2n minus 1, so it's going to be monosomy here, it's going to be trisomy here, normal diploid here, right? Diploid, trisomy, monosomy. Monosomy means only 1 when it should have 2. Trisomy means 3 when it should have 2. Same thing here. If this happens in meiosis 2 or in meiosis 1 here, where this tetrad doesn't come apart, so those homologous pairs stay together, more than likely they didn't have genetic recombination. There wasn't snip, 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 and share DNA. So they went, all right, let's just stay together. They stay together. Now they go under meiosis 2. Then they split apart. All the gametes that result are going to be trisomy. They're all going to be 3, because assuming they get fertilized by a normal sperm. Huh? So if we look at syndromes, some of these syndromes are really interesting. I have friends that actually, and people I know in my family and friends that have a lot of these different syndromes. Trisomy 21, this is Down syndrome. Okay? The karyotype actually has 3, hard to see, but 3 of chromosome 21. And that happened from non-disjunction. It could either happen in meiosis 1 or meiosis 2 of either sperm or egg meiosis. Okay, it could happen in either side. You have to do genetics to figure it out. But maternal or paternal non-disjunction in meiosis 1 or meiosis 2. So you have 3. And there are typical characteristics that go along with having this extra number, chromosome 21. She's adorable, right? And my friend, I should have got a picture of my best friend's sister, Down syndrome. She died at 37 though, so they typically have a shorter lifespan. They have increased rates of mortality and lower longevity. They have in general flatter facial features, kind of a flatter nasal bridge, lower indention in the nasal bridge. Medial epicanthic folds, this means little eyelid folds in the inner aspect of the eye. Speckled iris often, bulging tongue or kind of a protruding tongue, it's often an open mouth. These are sort of characteristics that go along with this. Now this is having an extra 21 chromosome. So there's more proteins that are coming and getting expressed from that third 21 chromosome. Normally you have 2, you have 3, you have excess. So this is coming down to sort of a dosage effect. We are set to have pairs, not a set of 3 or a triplet. Yeah.
So like, how does that happen in like the body? Like, it's just natural, or like?
It's not natural. It happens. It just happens. And when I get into pregnancy, you'll see that trisomy 21 is exponentially increased with female age, and it happens by non-disjunction in female meiosis, eggs in female meiosis. So trisomy 21 and miscarriage rates and even twins go up dramatically when a woman reaches about 35 to 37 years old. Yep. So we'll talk about that in pregnancy though. We'll come back to those things that happen, the risks that go along with pregnancy. This in general, it can just happen. It happens. Sometimes there's non-disjunction either in the egg or the sperm, and that gamete gets fertilized, and then you end up with a trisomic baby. And trisomy 21 happens to be one that the body doesn't naturally miscarry as often. So developmentally, it's able to develop and go all the way through to fetus and then be born. Yes?
Are you able to detect chromosomal non-disjunction while the fetus is in the womb?
Yes. We're going to look at that right now. Thank you. Perfect segue. So other syndromes, we're going to look at that in just a second. Syndromes that result from chromosomal non-disjunction, some other ones. 45X, or we call it monosomy X. Females should have 2 X's. This situation, they're missing the second X, so it's monosomy X or Turner syndrome. This happens about 1 in 2,000 live female births. These individuals have a shorter stature, they have other features, and this is one right from your textbook here. So they tend to have more of a shield-shaped chest, sort of a thicker neck, almost a club or a shield-shaped chest. Often they have this case, it was a very thick, almost webbed neck that they can undergo surgery to kind of fix that. They are born, typically they have learning and cognitive disabilities or delays in those areas. They have an absence of a menstrual cycle, and typically they have streak ovaries. That means the ovaries didn't even form properly. The ovaries didn't form properly in the fetus when they were in utero, and they didn't have meiosis of the eggs and everything properly, so they're infertile. They actually won't start menstruating. They have streak ovaries, and usually they have a very early loss of all eggs and infertility. They can have a loss of all eggs even before birth, before they are born. It depends on the severity of the situation in their ovaries. And they can take hormones and do all that and start cycling and stuff, but typically they do not have their own eggs, and so they are infertile. All they are missing is 1 X. So they have the 44 autosomes and just 1 X. They don't have the 2. So it's critical that females have 2 X's to have normal fertility and normal development. Another syndrome is XXY. You guys have probably heard of some of these, right? Some of these are typical ones. What I'm linking this to now is meiosis non-disjunction. These are just happening from meiotic problems when meiosis chromosomes don't disjoin, bless you, and they stay together. Klinefelter syndrome happens about 1 out of 600 males. So it's a little more common than some of these other ones. Slightly feminized features, underdeveloped testes and scrotum. Here's an example. This image is right from your book. There's a little bit of breast development, sort of feminized features, longer limbs, less body and facial hair, sometimes no facial hair or no body hair, very little. Breast development is known as gynecomastia. Gyneco is female, mastia, breast development. So there's some breast development in these individuals. Less muscle mass, so lower lean body mass, and typically taller. So they tend to grow taller and have longer limbs. Infertility often, or reduced sperm counts. So again, this individual, typically a male, has what sex chromosomes? XY. So in this individual, when this happens, they have non-disjunction of the X. So there's two X's with the Y, okay? So there's actually an additional X when there shouldn't be. And it results in this suite of features that we refer to as Klinefelter syndrome. These individuals, though, they identify as male. Often they just identify as male. They have slightly feminized features. There are some testosterone level situations, sometimes libido, reduced mood, depression often, especially things going along with fertility and a lot of those features that deal with that. Yeah, Bobby.
With the last slide that you were just on saying that they're missing one, is that the XO?
Yes, you can call that XO. You can call that XO or monosomy X or Turner syndrome. It's all the same thing. Yep.
So would these
Melissa.
people be considered intersex?
That's a good question. Most would not consider themselves that, no. Most individuals with Klinefelters, they are male, they feel masculine, they have traits, they have penis, they have features, they just often have some breast development and some slightly feminized features. They still have a deeper voice, they're still tall in size. I don't think I've ever met or I know of individuals that will really consider themselves intersex or non-male. Could be wrong, but in general, and I actually know some in my life. So, yeah, I need to carry on though, because there's more. There's another interesting one I want to make sure we get to. Klinefelters, okay, typically they identify as male. They have fertility. Oftentimes there's a case study. If you read in your textbook, this section comes right from the reading in your book. There's an individual that has Klinefelter and he talks about the difficulties that come along with it, and he never knew. He just knew something was off and sort of different, and things about his body and things about his mood and his feeling, and he was working his job and he realized and he got tested and found out he had Klinefelters. He had an extra X. He had never known his entire life. And that, just knowing that, allowed him to sort of make some changes and do things that made him feel better. So read the book, especially when it says like special focus. It goes over people's lives that have these syndromes and diseases and genetic things that I find really interesting, and that's why I like the book when it does that. So how can you test for this? I think you guys just said. You can test for genetic testing and chromosomal syndromes by doing either an amniocentesis. Yes, does this make sense? Are we good? Yes? Amniocentesis, which is done in about 12 to 16 weeks. Takes about 20 days for the genetic testing results to come back and be interpreted. You can also, from this, get information on alpha-fetoprotein or AFP levels, which indicate neural tube defects, NTDs. I'm putting abbreviations because this is all in your textbook, straight from your textbook. Some of the first stuff wasn't, but this is. You can also have done chorionic villi sampling, okay, or CVS, CVS, like the drugstore. CVS, chorionic villi sampling. You send in basically a little tube, a little aspiration tube. They shove it up through the cervix into the uterus, where you have chorionic villi, where the placenta is sort of developing. You can get tissue and cells from the chorionic villi, the little finger-like projections of the placenta, and get that tissue. Sometimes they can also do it by needle. They can go straight in with a needle and suction out some of the chorionic villi. This, you have to do this, you can do this earlier. You can do chorionic villi sampling in about 7 to 8 weeks. Takes 10 days for the genetic testing. This is higher risk in general to the fetus. It's very young, it's early, it's tiny, it's the size of a bean. So there are situations where the needle or the aspirating suction punctures the limb or a part of the embryo. Doesn't always, it's rare, and there's not a lot, there's controversial numbers and stuff on that, but there is a little bit higher risk. The thing is, this is done earlier, and the genetic results come back quicker. You have to wait 12 weeks till there's enough amniotic fluid to be able to do this and aspirate out some fluid. The fluid has sloughed off skin cells that have the DNA and the chromosomes from the baby, from the fetus in it. So this is later, takes longer to reveal. This is earlier, there's a slightly higher risk. Now I will tell you, when you hit 35, if you're having a baby around 35, you will almost be really prompted by the doctors to do one of these two mechanisms because of the risks of genetic and chromosomal syndromes. So they will say, if you really want to know, you should do amniocentesis or chorionic villi. Or you can say, no, that's okay, I'll just do the regular ultrasound and detect any measurements you can that way, because they can detect certain things like neural tube defects by ultrasound, which is benign. But these are sort of the common ones that are done to detect genetic diseases and chromosomal syndromes, like we talked about. Okay, I guess that's it. We'll finish the rest of this. We're going on to puberty and reproductive anatomy. So we're getting into all the sex stuff now. Now it's all about sex. And we'll start that on Friday. I'm going to play that song one of the lectures. One last thing. I have volunteers for fasting. Yes, for that tomorrow. Raise your hand. I have at least. Okay. Yes. Okay. I'll see you guys live tomorrow. Dress comfortable. Make sure your nutrition stuff is done, because I'll be checking over your questions and your data for that during lab.
Can I get a new?
Oh yeah. Let me give you a new card.
Thank you.
Okay, let's